Namaste and welcome you all in monthly KSAS talk where we invite the people from different disciplines and they share their thoughts. Today we have with us Dr. Tanushri Kaul, who is distinguished scientist and group leader at the International Center of Genetic Engineering and Biotechnology. She earned her PhD in botany from University of Delhi and is the leading expert in plant molecular biology, genome engineering, and bioinformatics, and as well as the stress biology. Using advanced CSISPR-based tools, she develops climate-resilient, nutrient-rich crops, and including iron and zinc-enhanced rich component a recipient of BioCare and RGYI awards. She is nominated for ICAR, AgriVision and VIWA 2025 award, Advancing Sustainable Agriculture and Food Security. So she will be delivering her lecture today on CSIR, SPRING, a path of climate resilient nutrient rich crops. So it's very much important for KSAS people and also for those who have interest in science and in field. So
CRISPR'ing. The word is CRISPR'ing. CRISPR is a process in genetics they use. CRISPR'ing a path to climate resilient and nutrient rich crops.
Okay.
Very important.
Okay. So welcome, ma'am. Please.
Thank you, Dr. Alok, and thank you, Dr. Balram. I take this opportunity on this day, March 8th, which is the Women's Day, and I congratulate and I celebrate the different women that have contributed to the progress of our country and the world. Some of them are those on whose shoulders we are standing, and then they have hand-holded us, and we are now in this position to think on our own and build our own economy in a way to develop our country towards Viksit Bharat by whatever 2047, whatever Modi ji's dream is. So I wish to contribute to his dream by developing. One second, what is this? Okay. So we at ICGB, I am Dr. Tanushri, and I am a group leader at ICGB, that the group which focuses on crop improvement with special focus on nutritional improvement of crops. So we work at the confluence of plant and human health, and our goal is to shift the focus from better feeding, which has been there for the past 70 years, towards better nutrition, which is the need of the hour. And our lab leverages the genomics and the CRISPR-based genome editing technology to redesign the crops for developing more nutritious crop that are more climate smart, that are more sustainable. So this is basically unlocking the power of genome editing to develop precision agriculture, crop-based, crop-specific, location specific and state specific. So this is what we aim at. And our group is mainly women scientists. Few of them are men, and I always respect the men that always are in the background, that give the freedom for the women to work so many hours to develop these lines. They are all young women with They are all harbingers of innovation by developing these genome-edited crops. So the lecture that I've been told for today is, for the KSS lecture series, is CRISPR'ing a path to climate-resilient and nutrient-rich crops. So CRISPR, as you know, has been evolved by two women scientists, one from UC Berkeley, Jennifer Doudna, and the other from She's from Max Planck now, and director of Max Planck plant pathogen microbe department. She's heading that. Her name is Emmanuel Charpentier. Both of them are had a common project which generally wanted to decipher how viruses attack bacteria. And this is through that project, a simple project, that is CRISPR, which is a local locus in the bacterial genome, that has eventually, evolved to, evolve as adaptive immunity against viruses. So the bacteria always take a piece of that viral genome and insert it into their in their own genome. And whenever the next time the same virus attacks the bacteria, it utilizes this vaccination card sort of a thing, which it remembers and then attacks back the virus, therefore making it dysfunctional. So all the lethal viruses, they control through this adaptive immunity. And the enzyme that was, functional or more magnificently working for this, it was a precise endonuclease, efficaciously and targeted way it used to edit these genomes, and that was the Cas9. And eventually later Cas12a also came up for plant genome editing. These two have been the most prominently efficacious tools for diverse crops. So Cas9 and Cas12 are the two endonucleases, and this strategy has been a welcome approach, because previously the GMO strategy have been, whatever GMOs have been developed in India, they all failed because of NGOs and activists like Vandana Shiva and her likes, where they just clearly say that it is harmful for human beings. So the GMOs did not see the day of the light, but eventually we now use this CRISPR technology to edit the native genes in the crops, so that we are not introducing any alien genes or trans genes or any marker genes or antibiotic genes. We're simply editing the native gene of the crop so that there is minimal modifications. So this is what today's topic is. I would be embarking on this journey. It would be a short journey, but it would be more of the results part and how the plants are now, in which stage they are. So you would be availing the flavor of the genome editing approach that we have done in our lab. So as of now, we all know that the Government of India has deregulated genome edited crops in April 2022, and they have set up a guideline. DBT is following up with these edited lines, and as of now, SDN1 and SDN2 have been deregulated, whereas the SDN3, which requires or which involves addition of new genetic material like big genes above in more than 20 base pairs or more than 1 kb. So all those aspects are under the SDN3, which is not allowed as of now, but eventually it may be. So these site-directed nuclease 1 and site-directed nuclease 2 we have developed in our lab, and these have different crucial traits for enhancing sustainability and productivity, as well as nutrition under stress conditions. So SDN1 basically involves knockout, and the repair is done through the endogenous mechanism of the plant. So here we are not introducing any DNA. We are just creating a knockout. That knockout or that mutation that is created is a double strand break that is created, is repaired by the endogenous machinery of the plant, and that leads to insertions and deletions from on the basis of whatever pathway it follows. Like if it follows the non-homologous end joining pathway, or it will have insertions and deletions. The second pathway, second kind of these side-directed nuclease 2 basically involve knockout of a piece of DNA and knock-in through homologous donor repair template mechanism, and this involves few base pair changes. So this goes through the homologous donor repair pathway, which is more error-free, and hence we have utilized both these. So the first product that I would wish to highlight or showcase is the development of dry direct seeded rice, which is the need of the hour. Why? Because this is the best alternative to the water guzzling conventional, traditional transplanted puddled rice. As you all know, that leads to a lot of global warming because it facilitates different methanotrophs that increase the production of methane. And also it requires a lot of fresh water. We know that almost 5,000 liters of fresh water goes into development of 1 kg of rough rice. So this is why the northern states of India like UP, Haryana, Punjab, and all these states have, their water table has gone down. Even NASA and ISRO have shown these pictures where the water table on these northern states has gone down. And hence the government of India is pushing or nudging the farmers to utilize dry direct seeded rice, which does not require water as much as it is required in the dry direct seed. So this is called like tar vatar. So it is whatever minimal water is required that will be given, but not the way which is traditional way of puddling and keeping transplanting into a from the nursery to the field with full of water and all. So that is how. But there are pros and cons of this technology. The pros are that it reduces water usage, it reduces methane emissions, it also ensures shorter crop duration, early maturity, better soil structure because the hard pan is not there. So there is so succeeding crop grown that are eventually the succeeding crops that are also improve, their yield also improves. However, there are different challenges that direct seeded rice has. Mainly the first is the weed rivalry, because the water was a natural herbicide and it used to curtail the herbs or the weeds. Now once the water is removed and only minimal water is given, so the weeds, numerous varieties or diverse weeds have cropped up. And also the second challenge is the micronutrient deficiency. There are other challenges also, you can see here, but the most crucial are the weed rivalry and the micronutrient deficiency, so which we have catered to. So we have chosen this glyphosate, which is a broad spectrum, non-selective systemic herbicide, although availed a bad name, but I would tell you how it has overcome the, it has changed, revolutionized the crop agriculture in different countries, and in India it's still not being used legally. And this is how the in the pathway, shikimic acid pathway, it attacks, or it competitively inhibits PEP, that is the second substrate of this enzyme EPSPS synthase, and it goes and binds into this in place of PEP. Hence, these three amino acids, the aromatic essential amino acids, are not produced, hence the weed dies. However, in the field conditions, the weeds and wild type both are affected, because the same pathway also happens in the main crop, in the rice or corn or whatever crop you're taking, because it is in the chloroplast, this EPSPS synthase, the shikimic part. So we need to protect our crop, and that we have done through introducing three mutations in the EPSPase gene by knocking out a piece of gene that is a conserved region and knocking in the desired mutations through homology donor repair mechanism. Hence, now the plant, the main crop does not die when you're spraying it once or twice or thrice. The herbicide, the plant does not die. Although the weeds die, but the plant resists the herbicide because now it becomes insensitive because of the modifications, and it also now is more robust and it suppresses the weeds better. It's not like the previous. And moreover, when you edit this, we also serendipitously found that the interaction between these two, PP and EPSPS, enhanced, so that led to increased aromatic amino acid production and higher yield in these crops. So this is how we edited the native gene. We introduced two guide RNAs flanking the conserved region, and we knocked out this piece of DNA, and we introduced a donor template with the desired mutations. So it was in place of G, it is A, in place of T, it is I, and in place of P, it is S. And eventually through site-specific homologous recombination, the left arm and the right arm is identified, and this similarity is achieved, and then the central part is shoved into the this native DNA. So this is how it happens. This is precise DNA modification, and this we have utilized leveraged in different crops. So I'm showing you for rice, but eventually we have done it for corn, for pigeon pea, for soybean, and we are now doing it for mustard. So this is how it modified, three modifications led to the insensitivity of glyphosate. And the cavity changes in the form that it was initially, it had more depth. Now, because of the modification, the depth is reduced and it becomes more open, which does not fit in, which is not suitable for the glyphosate structure to fit in. So then we have my student from Bangladesh. She developed these lines in the lab, and these lines were eventually, tested for transgene-free status. And see, the southern analysis shows that these were Cas9 free, because this is the transgene that we need to segregate for before we bring the plant into the field. And after Sanger sequencing, we identified that the three mutations were there. This is the wild type, which does not show the mutation, and these are all changed here. So these lines now have been developed, and they've been dossier. We've made the dossier for DBT, and we've sent it to IBSC for deregulation. There's a phenotypic analysis of the wild type. C is the control, and E is the edited line. So you can see that the robust root development is there. There is also elevated yield. Production was elevated by 20 to 22%, and there were no morphological or abnormalities or yield penalties as of in comparison to the wild type. So what is this? What I wanted to say is here, when the glyphosate we've edited and the glyphosate is not binding to EPS space, but serendipitously the PP is binding more strongly to EPS space. Hence the shikimate is being utilized in a better way, and the aromatic and essential aromatic amino acids have enhanced. So this we have checked through a shikimate analysis and the aromatic amino acid analysis, and you can see the enhanced phenylalanine, tyrosine, tryptophan in the edited lines, and the shikimate is better utilized. Say this is the control line, this is the treated control. So in the treated control, the enzyme is not working, but in the edited lines, the enzyme is working more better than the control lines. So it is utilizing the shikimate in a more better way, and hence enhanced aromatic amino acids are seen. So this is the picture which is showing the edited lines that we grew in the greenhouse in ICGV, where we wanted them to be sprayed with glyphosate, which is Roundup. And from Monsanto Bayer company, we got this, and we eventually performed the glyphosate foliar spray. So this is before spraying. So we utilize three events, 2, 6, and 9, and we see that it tolerates after 10 days post spraying. It tolerates, the edited lines tolerate different concentrations up to 1.5 ml of glyphosate ml per liter and 3 ml per liter of Roundup, basically. So these two lines now we have, for the dossier development of the DBT, according to the DBT guidelines, we are developing the dossier so that they get deregulated. These are the plants that were grown, but they were not sprayed with Roundup. These are control lines, wild type lines, and when you spray them, so at these two concentrations, on which the edited lines survived, these control lines died. These are the non-sprayed wild type lines. The second product that I would wish to highlight is the redesigning of rice crop for improved micronutrient quality in starchy endosperms. As you all know that in the polished grains and the milled grains, there is nothing much nutrient left in the endosperm except for 2 to 4 ppm of iron or 7 to 16 ppm of zinc. So this is a holy grail in the transgenic approach and the breeding approach, which we could not break. And through this genome editing approach, we introduced 3 gene mutations, which led to the enhancement of iron and zinc in the grain endosperms up to 25 ppm to 120 ppm of iron and 30 to 145 ppm of zinc. So this is an unprecedented increase in iron and zinc in the grain endosperm, a feat not achieved in the past either by crop breeding or transgenic approaches. So this is the slide which is highlighting the plants edited lines with high iron and zinc in the grains. So this is the dose stage and the mature stage. And these are the comparative panicles and the seeds. This is the wild type. The wild type. This is the wild type. The first one is the wild type, and the rest are the heritable types. So you see that these are Samba Masuri rice events, and we eventually did the agronomic trait analysis also, and the edited lines performed better. The blue one is a wild type. I'll show the picture again later. So these are the iron and zinc staining, and also cadmium. We also reduced the cadmium content. So this by utilizing two iron sensors, we knocked out two iron sensing genes that eventually, negative feedback to the roots to stop absorbing more iron, and thus the grains are having less iron, reduced iron. So if when these are knocked out, so there is no control, no negative feedback, but there are other genes that eventually come up and play their role in controlling that. But in the initial stages, which leads to less iron and zinc accumulation in the grain, these two iron sensing genes, once knocked out, led to enhancement. And plus we also knocked out this cadmium transporter, which is basically working as a transporter from xylem to phloem, from phloem to grain. 100% transfer of cadmium is done through this transporter. So once you knock that out, instead of cadmium, iron and zinc goes into it, into the grain, to manage the iron balance. So this is the mice studies that we have done to show the histopathological studies showing that there is no toxicity due to this grain, feeding. We did the feeding trials. So this is all. So here again, you can see here, this is the pearl Prussian blue staining, where you can see the central part. This is the wild type. This column, first column, is the wild type, and the rest are the edited lines. You see the central part is now rich in iron, stained by pearl Prussian blue. And this is the central part which is stained by RTZ for zinc. So there is enhanced zinc and iron. And the cadmium content decreased. This is the dye staining the cadmium content. So this is how the stereomicrographs reveal that the iron and zinc and low cadmium, high iron, zinc and low cadmium in endosperms of knocked out mutant lines. This is visual representation. Again, we also show that in contrast to the wild type, the edited lines exhibited profuse root growth and shoot growth. This is under iron sufficiency, and under iron deficiency also. Whatever is there in the soil, these plants will absorb and bring it into the plant and the seed. So this is after 21 days of growth in the hydroponic media. So again, the wild type and edited Cas9 lines, you could see different grown in different iron and zinc concentrations. So you can see that the edited lines tolerate up to 200 micromolar iron and 650 micromolar zinc, whereas the control lines give up at 100 micromolar zinc and 50 micromolar iron. Similarly, we grew the lines along with the wild type on different concentrations of cadmium, and here you can see that the edited lines could tolerate up to a maximum up to 650 micromolar. After that, though, the control lines gave up at 100 micromolar. They could not grow after that. So, moreover, the genome edited high iron zinc content also led to increased, flowering, and it also led to early accelerated maturity. Early flowering and accelerated maturity, like 15 to 20 days earlier than the wild type lines. These lines, edited lines, could be harvested. So you see these are still, they are pollinating, and here you have already the seeds are being set. So the question arises whether this high iron and zinc is creating any reactive oxygen species in the leaves or in the grains. So we tested it through histochemical detection of ROS we wanted to see. So through a peroxide, hydrogen peroxide, and O2, that is, we wanted to test. So we used those stains to detect these. That is NBT. And eventually, we saw that the edited lines did not show any bronzing. There's no toxicity in the leaves or grains as compared to the wild type. So eventually, in addition to high accumulation of iron and zinc in the seeds, the 3 gene mutants of rice also showed enhanced nitrogen uptake. So I found some data. I think those lines are data is missing, but nevertheless, I want to say that the different concentrations of other micronutrients and macronutrients were tested, and these were found enhanced seed, like magnesium, calcium, potassium, phosphorus. They were significantly higher than the wild type in the seed. So not just iron and zinc, the other micronutrients and macronutrients also were absorbed efficiently in these edited lines. And then when we checked the nitrogen translocation, what is the situation? So we found that the edited shoot showed enhanced nitrogen uptake in comparison to the wild type shoot. And also the cadmium and the cobalt content were analyzed in the shoots. You see the cadmium and the cobalt is absorbed and it is stored in the root, edited root, as compared to the wild type. So the edited shoots do not have cadmium or cobalt accumulation. So eventually we could make this model where we show that the iron and the zinc from the soil is accumulating in the endosperms of the grain, whereas the nitrates are going towards the biomass, and this leads to biofortification, whereas the cobalt and the cadmium are withheld at the root level. Hence, it's effectuating a bioremediation kind of an effect. So these are the comparative morphological, agromorphological trait performance analysis. The wild type line is in the blue color, whereas the edited lines you can see in the yellow. So you can compare the different traits like flag leaf width, total yield, 50% days to 50% flowering, primary panicle branching, secondary panicle branching. So all these traits were exhibited enhanced growth in all these traits. The edited lines were performing better than the wild. You see the 100,000 seed weight, the plant height, tiller numbers, number of grains per panicle, and so on. And this is the Cas9 free lines, the 8 Cas9 free lines that we now have developed the dossier and we have delivered to the IBSC. So this is again the iron, zinc, and cadmium staining. As compared to the control, you can see it's higher. And so here, what I was saying is, with this gold standard analysis of ICP-MS analysis of iron, zinc, cadmium, so you see that even cobalt and cadmium in the seeds is less. So this is the control line, and this is the cadmium, and this is the cobalt. So these are the edited lines. You can see, although it is very less in the control, although it's very less, it's 0.01, but even in edited line, it's even lesser than that. It's almost minimal, negligible. So there is no accumulation in the grains. Whereas the zinc and iron, you can see up to 140, 145, and iron up to 120. So this is basically the figure showing how the other nutrients are working. That's what I was saying, that manganese, potassium, phosphorus, magnesium, copper, calcium. So this is a control, the right, and the other lines are showing you the edited lines having significantly higher potassium, magnesium, manganese, phosphorus, calcium, copper levels. So it's like a micronutrient capsule. The rice grain becomes like a micronutrient capsule. So this here slide, I am shifting my gears and showing you how DNF-free RNP mediated approach could be used for editing different crops, and it is important for those crops like saffron and turmeric and banana, sugarcane, which are basically asexually reproduced, or they go via vegetative reproduction. So for saffron, we had to standardize this because we also intend to work on saffron, and we have eventually received a grant recently, but I'm not showing that data. So for that, we developed this protocol, and we tried to extrapolate it to rice, and we saw that when you use a 2 to 3 day embryo of rice. Once you germinate the seeds and then you isolate the embryos from there, these embryos 2 to 3 days are very sensitive. You can use them for uh, editing. So we directly shot the guide RNAs and the um, whatever the of different genes. For example, we started with the phytoin desaturase. So you see that it leads to reduced chlorophyll accumulation and the growth is reduced. Then we did it for dense erect panicle 1, which is basically for increasing the panicle number, for increasing the yield in rice. So you can see that the number of panicles increase. This is the wild type. And this is the edited DEP1 mutant. And the third one is the 1000 grain weight 6. So in addition to DEP1, we added another editing in this same line. So it's like a DEP1 plus 1000 grain weight 6 together, because in this case the DEP1 had a drawback, which is simply is that the culm of the plant is not very strong. So to increase the culm strength, we introduced the knockout of this gene. Hence, the DEP1 plus TGW6 gives you higher number of panicles as well as a stronger culm and more robust root growth. So this is what is highlighted here. So we got the genome editing efficiency up to 85% approximately. So this is how we shifted to we can shift towards DNA free technology. So this is again the extrapolation of the rice, whatever we've done in rice for editing through homology donor repair mechanism. For rice, we extrapolated to corn and pigeon pea and also soybean, and we have a patent for all these. So basically we efficiently modified that PEP binding domain in the EPS space that I already showed you. So this is again the same pathway that we targeted in the rice. We did it in corn. We have publications for all these papers are published. You can I can send you the papers through chat or through Alok. I can send anyone who requires. These are the corn lines that are SDN2 lines, glyphosate tolerant lines in the greenhouse. So this is how we again utilize the same strategy. Homologous DNA repair mechanism, where precise sequence modification, error-free modification can be achieved. And this is a very difficult strategy, not being done in India. Only our lab has been successful in this, and we have at least 3 papers on this technology. And this is very innovative and very clean off-target free technology. So there are no off-targets anyway. So that's why this strategy has been very difficult for Indian scientists. None of them as of now have achieved this feat. So this is for again we tried growing these edited lines in glyphosate media from Sigma, the simple pure glyphosate media, and phosphonomethane glycine. So that is 4 millimolar of that. So edited lines could grow on it, but the wild type could not grow on 4 millimolar glyphosate. So this is basically the T1 lines, T1 seeds when we checked for Cas9. So these are the Cas9 positive lines. Those that showed Cas9 positive PCRs were tested for the gene sequence PCRs. So we screened those gene PCRs and from nested primers 1 and 2, and then eventually we gave for sequencing, and then we found around 13 lines we found which showed 3 mutations in this corn EPS space native gene. And here you could see after glyphosate Roundup spray of these lines, these are control lines which could not recover properly. So this is up to 6 ml, but that's why it is too high for these lines. But our edited lines could survive up to 6 ml per liter. So these are the T1 generation edited lines in the greenhouse. And these are the T2 lines. So every plant gave one cob. They were then checked for the photocentric parameters through infrared gas analysis. So this is the control line, which is not treated with glyphosate, but this is a control line treated with glyphosate. And now you compare this in the field with the edited lines, which are treated with glyphosate. So the edited lines, once treated with glyphosate, perform significantly better than the treated control. So it clearly this can be seen and visualized that when you spray glyphosate in the field, your main crop definitely is affected so much. Sometimes it doesn't recover, and it may take more than 15 days to 45 days to recover, and sometimes they do not recover. So this is the EPS-based activity that we checked in the edited lines. So these are dose dependent. To determine the EPS activity, which was based on the inorganic phosphate release, was measured by malachite green dye-based assay, and this is the different lines, 7 important lines that we chose, and this is the control line without glyphosate, and this is a treated control line. So they are significantly better than treatment. So again, the shikimate was analyzed in the edited lines. You see, it is better utilizing the shikimate here, like in the rice, the native EPSPS of corn also working, edited EPSPS of corn, the native gene working well, absorbing or utilizing shikimate very well as compared to the untreated control. But this is the treated control where the EPSPS is not functional because the glyphosate binds to the EPSPS and makes it ineffective. Hence, it is not able to utilize the shikimate. So this is again the aromatic amino acid analysis in the corn, where you see the control. The blue is this the control. You see in the other these other treated edited lines M1, M2, M5. So all these aromatic amino acids are enhanced here. So these are the comparative analysis of the edited mutant cods. So you can see there are no defects in the size of the kernel, no dislocation, no edge dislocation, no abnormalities in the ears. So there are no developmental defects. So eventually, once you keep on spraying the same herbicide time in and time out, again and again every season, the resistant weed starts coming up. So this is the one resistant weed that has developed by spraying for several years. So eventually, slowly, you get more resistant weeds. Time in and time out, every season, if you spray the same inhibitor or the same herbicide. So what we so this is threat to herbicide-based weed management. And also you can see in BT cotton also you can see the same situation where the uh, pink bollworm and the fall worm, they are resistant to BT as of now. They are eating the BT cotton because we are not giving any refuge crop. We are not growing any non-BT along with it. So the crop, the insect is so intelligent, it adapts to the BT, and now it's surviving. So we need to have a strategy where multiple herbicide resilience is there in the crops. So that is what my aim is. I have been doing it since several years now, because several years in the sense that we have developed these lines in 23, 24, and these are now. This is a new project where the company has partnering us for developing multiple herbicide resilient corn. And in this case, we are using ALS astrolactate synthase mutations also along the EPSPS. So again, the EPSPS will be mutated. Along with that, the ALS also, astrolactate synthase, which is the enzyme that, produces the branch chain amino acids. So these are also essential amino acids. So along with EPSPS, together we can, for 1 year or 2 years, we can label the batches as for glyphosate, and the next third and fourth year we can label the seeds as imazeth hyper resistant or any other bisparax sodium resistant. So there are several herbicide resiliences that we can introduce through ALS. So these two mutations have led to resistance to 4 to 5 different herbicides in addition to glyphosate. So this we have doing it in maize. Now we've already done it in pigeon pea. So our PhD student Jyotsna and another postdoc Rachna both have developed these lines. These are double herbicide resilient. I mean double genes, 2 genes have been targeting this. So here they have introduced 4 mutations in the EPSPase, native EPSPase gene, not introducing any transgene. The native gene has been edited to show 4 mutations, which would lead to enhanced glyphosate resilience, and the ALS mutations, 2 mutations, P2S and W2L. So they are in the second gene. So these lines, so this has led to the resilience to, uh, uh, 5 non-selective pre and post-emergent herbicides in addition to glyphosate, which is a post-emergent herbicide. Many of these are pre-emergent, like imazathioper, bispirac. These are pre-emergent also, and they work as post-emergent also. So you see the wild type seeds are not being able to grow in these herbicides, but the two edited lines, two events that we have taken up, they are growing in the different concentrations mentioned here in these different herbicides. When they are transferred to the pot, but they still do not survive the wild type, not happily survive. But these edited lines are surviving because the concentrations are at a very higher end. So these wild type are not surviving. So we have resistance to glyphosate, imazathioper, chlorsulfuron, bispirac sodium, chloransulam methyl, and fluorocarbazole sodium. So many of we can use alternatively so that resistant weeds do not develop. So here these are the edited lines in the three figures, and these are the wild type lines which died after spraying. This is 15 days post spraying, foliar spraying of Roundup. And this is 30 days after recovery phase. So the wild types do not recover at all because the concentration is very high. It's about 3.5 ml per liter and 4.5 ml per liter. So even the aromatic amino acids are enhanced in the edited lines. So this is a plus point. In addition to giving herbicide resilience, there is increase in aromatic amino acids, and this helps us to boost, because these are all amino acids like in phenylalanine is a precursor which eventually leads to the production of secondary metabolites like phenylpropanoids, flavonoids, lignins, and oxins, and strigolactone. So these give you crucial for defense and stress adaptation. So they give you enhanced stress resilience also, in addition to herbicide resilience. So similarly for other inhibitors, we checked what happened in the edited lines after spraying, 15, 21 days after spraying, what happens. So we see there is enhanced aromatic amino acids and branched chain amino acids, because these are ALS. So this is branched chain amino acid analysis. So that is valine, isoleucine, and isoleucine, leucine, and valine. So this is for these other herbicides like triazolopyrimidines and fluorocarbazole. So this is again we extrapolated these same results for multiple herbicide resistance in soybean. We've started with EPSPS, 4 gene mutation, 4 mutations we introduced, and we are now beginning for astrolactate synthase. There are 3 astrolactate synthase genes in soybean. So it's a difficult ball game, a different ball game here. So we need to introduce the mutations, all the 3 isoforms of ALS, in addition to 1 isoform of EPSPS. So we've done it in one of the genes in ALS and EPSPS, but we are doing it for the other 2 genes of ALS. In this case, soybean. Again, another PhD student working on this. So there is another legume-based research, because it's a legume-based research. So I wanted to show her that there is this rice bean, which is an underutilized crop. We are exploring it because it's a very nutritious crop, high protein, high calcium, high vitamin levels, and high iron, high zinc. But it is not grown. It's not eaten. Not only northeastern parts generally grow them, but it is there in all the countries throughout the globe. Even US, Nepal, Australia, everyone has this plant. Africa, but they do not use it for human consumption because of its bitterness. So there are certain trypsin inhibitors that are creating bitterness, and there are certain anti-hematogglutinin that create some bitter taste. So people do not want to eat it. So even the animals do not want to eat it. So through genome editing, you would identify certain target genes that, when knocked out, can improve the palatability and also enhance the early flowering. Because in the the hilly regions, the flowering is late, more biomass is there, flowering is late, and it becomes like a huge crop. It takes 4 to 6 months, and after that flowering happens. So modifying the flowering genes, editing the flowering genes to knocking out the negative regulator of flowering genes, so that we can have biomass as well as early flowering. So this is how the lady called Pooja Chakravarti, she's edited. This is the wild type rice bean plant. It endlessly, it's indeterminate. It keeps on growing without flowering for 6 months and even more. So here she's edited, and there's an edited line where the length is less than the wild type, but the biomass is not compromised and flowering is enhanced. So this is what we are doing. And now one more last, but not the least, I will finish with cotton. So this is the plant which is most susceptible, most talked about, and most important, because as of now, India was supposed to be the one of the largest producers of cotton besides US and China and other countries. So it is under difficult situation because Bolgod 2 eventually is not very efficacious. But now Monsanto Bayer has come up with Bolgard 3, but that is only for those lepidopteran insects. So what we try to, develop we have a project on this from DBT. So we're trying to increase the fiber quality and the insect resistance. So as of now, we have tried to increase the fiber quality, where we have seen that the edited lines, these are the 3 edited lines, in comparison to the wild type, they are having more biomass, more flowers, more flowering, early flowering, as well as a better architecture. And you see here, the bowl size also is enhanced. This is the wild type. And you see the fiber length also is almost double the wild type. So you see the flower size also is more, the leaf size is also more. So this is how we try to edit a phytochrome based gene, and knocking out that led to this effect. So we are now patenting it for India. And this is again, you see the early flowering stage, you see the wild type and the knocked out plant, early flowering stage. And this is the bowl formation stage. This is the early flowering stage, the early bowl formation stage, wild type and knockout. And this is the later bowl formation stage. You see the size of the knockout bowl is also bigger. And you see the flowers are also bigger than the wild type. The leaf is also bigger than the wild type. So you see there are developmental changes associated with the phytochrome, that we knocked out a phytochrome related gene. So that led to enhanced early flowering and noticeable leaf size differences and flower size differences and bowl size, as well as the fiber quality. So the fiber quality you see here is, here you can clearly see under the scanning electron microscope, you see the wild type flower fiber. It shows a twist here, but when you compare it with the edited lines, you see there are more twists, and this is what the company looks for. This is the tensile strength, which is required by the companies, and this is what we look for. And the yield is higher, and strength is also higher. So this is again the edited line here, you see. Compared to, there are more flowers. And here you see the wild type. Here again you see the early flowering and bold formation in the knockout lines. And this is the late flowering and the late bold formation in the wild type lines. So basically it is basically a genome edited lines exhibited early flowering and accelerated bold formation compared to more yield. We checked through the real-time quantitative PCR analysis of this phytochrome gene, and we saw that in different tissues like the leaf, the root, the sepal, and the cotton boll, this gene was reduced. Expression was highly reduced as compared to the control. And then we checked the fiber length, fiber strength, fiber uniformity index, and cellulose content. So you see the edited lines perform better in all aspects. This is the T2 generation now we have. Now further what we are going to do is, these are the two important, the pink bollworm and the Helicobacter armigera, that are mainly affecting the bolls. But these are the lepidopteran insects, but there are other insects that are sap-sucking insects like whiteflies, mealybugs, and aphids. So whatever Monsanto Bayer has come up with, the Bolgard 3, that is not resilient to wildflies, mealybugs, and aphids. Resilient to pink bollworm and this Helicobacter, old bollworm. So these two. So the rest of the three are still, creating havoc in cotton. So what we are doing it in this project is we are trying to knock out certain target genes that would enhance the salicylic acid production, that would enhance the terpenoids and phenolics and alkaloids, as well as increase the trichome density. This is a gene multiplexing of several genes that would enhance all these aspects and would give very crucial edited lines that would be with enhanced yield. They will be resilient to different insects, including the sap-sucking insects like white flies, mealybugs, and aphids, in addition to pink bollworm and the other fallworm. And hence, this is what our target is. And then we have also worked upon reducing the allergenicity of tomato, like 14% to 20% of the population are allergenic to prophyllin in tomatoes. So we try to reduce the prophyllin and increase the lycopene content, regenerating. So this is what I end up with. And this is my team. These are different students that work with us. These are the previous ones, and these are the new ones. And these are different member state trainees that come, because ICGV has a mandate to train trainees from 70 different countries throughout the world. So the Nigerian Victor and Rita had come, and we trained them with the RNP strategy, the DNA-free strategy to edit, because their interest was cassava. So that is a asexually grown crop, vegetatively grown crop. So we trained them in. So thank you for your patience. And if there are any questions, I'll be happy to take.
Thank you, ma'am. Thank you for this insightful lecture. As we all know that India is Krishi Pradhan Desh, and it is said from the beginning, and very large part of population depends on agriculture. In recent year, we have also witnessed the farmer protest in India, and this also, established that the agriculture is the central part for economy as well as for, social stability. So, in this context, technologies like the CRISPR technology, which you beautifully explained, it aims to develop the climate resilient and the nutrition rich crop. It can play an important role and supporting farmers and also improving the productivity, which is the crucial part of agriculture, and also ensuring the food security for future for India. So thank you very much. Before anything else, I will invite Professor Balram Singh sir for his comment. Then we will take few questions.
Very interesting, very elaborate and extensive work. It's so amazing that you have done so much on so many crops. There are some questions I have. Is that you make these CRISPR based changes in the genetics. Normally, you change someplace, it has many other implications. But your data is so very precise. Have you really looked at that the implication of these genetic changes into other physiological or biochemical pathways?
So thank you, Professor Vardhan. Intriguingly, we also were skeptical initially, but when we did the transcriptome analysis and we sequenced the whole genome of these lines, we did not find any off targets or any mutations in the other pathways. We studied the ontogeny also. So we did not find any off targets because this is so precise and targeted. And mainly,
yeah, I'm not asking about the changing the gene. Gene technology is very targeted, but physiological implications when you change aromatic amino acids or whatever. I mean, there are so many roles that they have in basic physiology. Do they change any do they cause any physiological changes rather than the assumption that you have that some certain types of metals or aromatic amino acids or any other compounds you target, they can increase, but does that increase also change other pathways in the physiology?
So what I understood from your question is, yes, we did The answer is yes, we tried to see the physiology in the field, and we found that the edited lines were better, phenotypically better than the control lines. So they were more for example, in pigeon pea, they were more drought resilient. And the aromatic amino acids eventually gave a better resilience to other disease, pests like white flies. Generally, the control lines used to be affected by white flies in the field and in pigeon pea as well as in cotton. So even in rice, they sometimes come up. But in our edited lines, they could not survive. They did not affect. They did not habitat. I mean, they did not uh, create a habitat there. They did not sit on those leaves. So they were like, I think, some resilience of some kind, which is basically maybe because of the enhanced salicylic acid content that may have been affected by the enhanced amino acids, because I see the aromatic amino acids are precursors for other downstream metabolite, secondary metabolite synthesis. So when the PAL is increased, so eventually maybe the other enzymes are also increased, but we now are going to test, because this is a very recent analysis that we saw in the field, because our director wanted us to grow them and see them in the field itself. So we did it in the net house, and now this difference, and now we're going to analyze that what is the enhanced pathway or the metabolite that is enhanced. Maybe it's a saponin, or it's a secondary metabolic pathway which is affected in a better way, rather than the wild type, which is susceptible to white flies. This is not susceptible. So you're right. I mean, this is what we've identified recently. And for other crops, we haven't yet seen any negative effect in physiology. But I don't know. We have tested up to T3. Maybe we'll go further to T3, T4, and T5 and see. But as of now, we haven't yet seen any harmful effect, so as to say.
Yeah, I mean, the targeted questions, I think it's beautifully done. Whatever you have targeted for resistance, that looks like really very nicely done. And nutrient increase in the nutrient content also has been done. But like as you see, and you may have to do that in the future, we never know what is the nature versus artificial changes. I mean, we are also kind of in a way part of nature. So whatever our thoughts are coming is also kind of nature in some way. But then it will have its own consequences. We are making some changes in the nature, and nature had, like you're saying, that insects. India has the largest amount of variety of even insects, believe it or not. So they will also adapt to certain degree, and the question is how can we use them the best for our purpose, in terms of food scarcity or food nutrients. That's a lot of other study needs to be done, which is okay. That's part of the life. But I also wanted to ask another question, is that, you talked about rice or maybe other crops. But the crops that have been millet type of crops that now government is also trying to enhance. Have you looked any in those? Because those are like very natural types of crops and has grown over thousands and thousands of years, and humans have adopted. So have you really looked into that kind of things rather than just rice, that's something that we like?
Yes, that is very true. So we have been now working on Citeria, finger millet, and sorghum for the past, and Penicillium we had been working for the past maybe 20 years, but with a transgenic approach. But now with the millet year, we have picked up Penicillium, finger millet, sorghum, and Citeria. And sorghum particularly for increasing the striga resilience, and the other millets for higher yield and high iron and zinc content. So we have embarked upon the working on these millets. Yes, for penicillin we had been doing it because Dr. Reddy was my mentor. Professor M.K. Reddy was my mentor at ICGB, so we had worked with him for penicillin, to develop, to transfer the genes from penicillin to rice to make it C4 like, but then that was a dream not yet fulfilled. Eventually we for the when the genome editing thing came up, so now recently for the Gates Foundation funding, we have to work on legumes and millets. So we have picked up these four millets. Yes, we will be enhancing the iron and zinc and the protein content to cater to the protein energy, malnutrition, and anemia, and this stunting of children below 5 or even for adults. We are targeting that. And striga resilience. We have picked up certain target genes for enhancing the striga resilience. We need to reduce that parasitic weed. And also for brassica, we are trying to do for orobranchi. We are trying to develop certain lines, and also oil content in brassica that is not being enhanced by breeding strategies. So the scientists at ICR, they say that the oil content has now come to us plateau, and breeding they do not have the germplasm to enhance in Ginsea more oil content. So that we have targeted three genes to enhance the oil content in ginseng. These are the future crops. And saffron. So saffron, we are trying to increase the number of flowers per column so as to increase the spice, because the spice in it is the stigma, the three stigmas inside the flower. So if we have more flowers per column, we can increase the quantity of the spice. For India, it is important. So we are.
Thank you. Thank you. One more query still continues. Very, very interesting lecture. So I really enjoyed it. And I'm so impressed that you have accomplished so much from India and then also helping other countries, including Africa. India has a physics concept of system and surrounding. Well, it's not India, it's just a physics in general. You have to have a system and everything else is surrounding. And then there is a chemistry concept of equilibrium. You have to have a reversible process. Everything is affecting everything. And so it's like infinite. I mean, our lifetime, we have to take birth several times to be able to check that all out, as you can see your plans. But there is a philosophical, there are a couple of people here from philosophy, including Alok, where we call Aham Brahmasmi. And that the Aham and Brahm are obviously different. That's why we call it Aham Brahmasmi. It has to go through a process. That process is that of interactions. So in your experiments, you have done CRISPR-based genetic changes and maybe some affecting insects and maybe some plants which are acquiring some herbicide resistance or not acquiring. But we are talking about, at least hundreds and thousands of different other things that will take probably long time to reach and then affect, but they affect each other. So from philosophical point and from physics point of view, the repercussion of what you are doing will take long time. And it is what you are doing right now is you are addressing a question of nutrition and resistant to herbicides so that you can get a certain crop. But then implication of all this is going to be very long term. Have you ever thought about it?
Very beautiful question. And yes, I do read about physics and its different aspects that have come up recently. The quantum entanglement, for example, that any particle that is here affects any change in this particle here can affect the particle anywhere at the edge of the universe. So that quantum entanglement is there, and definitely we are all connected through the energies, through consciousness, whatever we call it. And whatever we do, whatever steps we take, or whatever changes we make as of now, maybe via innovations of genome editing or whatever, GMOs or breeding, all of them are on the same platter. So, for example, if you say breeding, if you take plant breeding, cross breeding, and all, so they also had their own positive and negative effects. Everything has its pros and cons. Everything will affect the other aspect. For example, the wheat that we eat now these days, that is 70 years after the previous wheat that we used to eat 70 years back, that had less of amylopectin A, less of glutenins and whatever you say, the wheat has now. What is this? Gliadins and gluten. But after 70 years of breeding, we have looking at only one trait, that is basically the biomass and the yield. So
That's Alan Chale.
that has led to increase in starch in the grains. The number of grains have increased, but also it has increased glutenin, gliadins, amylopectin A, which eventually the heart surgeons say all of these. Wheat is, out of all the plants now, wheat is the most dangerous because it goes and clogs your arteries. The amylopectin A, the gluten and gliadin goes and clogs your arteries. So we cannot isolate any change. It's not just genome editing. You talk about everything. Even if you speak about Iran and Israel war, if we think if we are in India, we are not going to be affected. Definitely, we are going to be affected by that. So everything is quantum entangled. Everything is interconnected. So we need to, think precisely and see what are the repercussions, and then move ahead with that. Definitely, that is true. We should. So what do you say?
All right. Well, Alok, you gave me so much time, and I have occupied the whole question answer session. But because I really like the discussion, and I like to continue further, maybe some interaction with Dr. Kaul, because we have this. Maybe I mean you're only presenting online, but if did you come to our place?
No, I haven't. I lost the
Sudhir
document.
Sapori has come a couple of times.
So next year I
So
definitely love to
Next
come.
year she
please
is
come and
coming.
see.
Yes, she missed.
Yeah, we have done some. We are doing something on this margin because we collected water from Kumbh, and then trying to understand the
Yeah.
microbiota, and as its effect on human beings and all kind of stuff. So we are at the cusp of science and philosophy, and as we don't emphasize enough that we have a degree of doctor of philosophy. So we don't have a degree of doctor of science. So we really need to appreciate the philosophy aspect of it. And maybe India being such an ancient country and philosophically so advanced in many ways, demonstrating that quantum entanglement you're talking about through rishis and munis, we may not agree today, but there is at least those kind of mentioning in the texts, in granthas, and as well as, I mean, some people still claim that they can do it. So we need to study the science of it. But anyway, I took so much time, Alok. In case other people have questions, please let them get the chance.
Thank you, sir. We have two questions,
Yeah.
Dr. Abhinandan Madi Tripathi and Dr. Manish Singh Kaushik. Yeah, please.
Yes, ma'am. So I have a question, ma'am. Thank you, ma'am. Very nice talk. I enjoyed it. Actually, I have a question. Most of the time, you use you making knockout and knockdown line. Suppose that sometime most of the character is governed by overexpression line. So how can we use the CRISPR to for making the overexpression line?
So there are different tools available.
Yeah.
This is called CRISPR-I and CRISPR-A. So CRISPR-I is the interference thing where you can reduce the expression of a gene, and the other one is where you can increase the expression of a gene by designing the guide RNAs in the promoter of that gene. So, and those modifications in the promoters that you can do for that gene will eventually affect its expression level. So that has been done. We have also been doing it. We have a project now on enhancing vitamin A, where vitamin A, this beta carotene, which initially the starting point of that is the GGPP, and that is not present in the first enzyme that will convert that GGPP to lycopene. That those few enzymes that are not present in the grain itself. So what we are doing is to activate those genes in the grain by editing the promoter. So you can do the promoter engineering through CRISPR genome editing.
Okay, okay. Thank you, ma'am.
Yes.
Yes, Dr. Manish, please.
Okay, so first of all, I would say, ma'am, thank you for such a beautiful talk. And while listening to your presentation, most of the question which I had in my mind previously has been already answered, and few of them has been answered while the discussion between you and Professor Singh. Uh, like the development of herbicide-resistant weed and metabolic trade-offs and off-target effects while generating the GR crop strains. But I have two questions, ma'am. That while studying the GR crops, have you seen their response in uh, other drought conditions or some nutrient deficient condition? So, what are the impact of other stress condition on these genetically modified crops?
So my lines are not genetically modified.
Okay, so okay.
As the CRISPR technology is the beauty of that CRISPR technology is that it does not introduce any alien genes. No transgenes are
Okay.
in.
Mm-hmm.
So the effect of the crop, whatever you're asking, it would be definitely as good as the effect on the wild type. But there
Okay.
would be a specific trait that would be changed. For example, one gene, the native gene sequence, if instead of G there is A, okay,
Mm-hmm.
and instead of T, what is this? What is the T?
Thymine, thymine.
Oh no, the other one. Uh, three,
Tyrosine.
Right.
No.
so threonine. So instead of glycine,
Threonine. Okay.
I
Right.
changed it to alanine.
Mhm. Right.
Threonine to isoleucine, and then proline to serine. So these
Mhm.
three mutations are creating steric hindrance, right? There is a
Right.
steric hindrance, as per physics, there is a steric hindrance that is being created, and that spatial interference would eventually, what has it has resulted in is it is not binding to the glyphosate molecule. Although the
Okay.
glyphosate molecule and the phosphoenol pyruvate molecule both are structurally analogous, similar, very similar,
Mhm. Mhm.
and hence the glyphosate is, has this capability to replace it and inhibit PEP. But when you change this steric hindrance is created, okay, that cavity was initially a deep cavity, and now it has become an open, less deep cavity by these three mutations. So it
Mhm.
has definitely created a steric hindrance, and there is a vibrational stability that has been achieved. But this has led to a better trait. That means the PEP is now binding more efficaciously to EPSPS rather than previously in the wild type, and also the glyphosate is no more able to bind to EPSPase enzyme, which previously used to affect the EPSPase and the amino acid production. So now the plant edited line, which is the genome edited line through CRISPR, you edited the native gene in such a way that the enzyme is now acting more efficaciously than the wild type enzyme, and it is also insensitive to glyphosate. So what is the benefit is this, so that it's not a GMO. So depending upon what you say is drought and all, that would be assessed when it goes to multi-location field trials, which is now
Okay.
we're going to undertake. So that
Okay.
if trials, the for the rice at least, the IIRR will do. And we have also a partner, Nath Bioseeds, Nath Biogenes. They are also going to take these seeds after this deregulation. So both of them will do that growing in multi-location and seeing what is happening in different locations. And different locations definitely will be different stress conditions, different temperature conditions, different rain quantity. Right. So that is after those tests we could eventually
Okay.
say.
Okay. Thank you. And one more question, ma'am. Uh, in case of this CRISPR-Cas studies, so there is one more problem that the low SDR efficiencies.
Low.
So the high precision repair system, what we call it, I think. There is
Ah,
one terminology as
yeah.
DRF chances, which is very low in case of this CRISPR-Cas gene editing technology. So I just wanted to can you please shed some light on this part also?
So it is not uh, based on CRISPR. It is basically the plant does not uh, does not choose HDR. The choice of HDR is less than the choice of non-homologous end joining. So
Right,
you get error-prone editing. But sometimes that error-prone editing is good because it creates frame shifts or point mutations, and hence the gene becomes non-functional, the protein becomes inactive, and hence the
right,
trait arises because then the endogenous machinery repairs it with insertions and deletions, which leads to these frame shifts, and then the different trait is created. But for homology donor repair, though the percentage is less, but by modifying the protocol, you can enhance it. For example, in our case, we used Cas9, which is a blunt end cutter, double strand blunt end cutter, which
mm,
which many papers will say that it is not very suitable for homologous donor repair. But it is in our hands. The protocol was modified in such a way that the homology donor template that you give, that should be in a higher quantity, and it should be given simultaneously so that once the knockout is there, that HDR has to be there, the template has to be there so that the plant immediately recognizes that and utilizes it. This is a technology, in 2007, Mario Capicchi from Italy, that he NIH scientist, Mario Capicchi, Italian scientist, he discovered this. It's called gene targeting. So, and to enhance these, he did it in the embryonic stem cells of mice and yeast, and he utilized certain marker genes to see the efficacy. But in our case, we cannot use marker genes because we have to, go through the IBSC. So we cannot create GMOs anymore. So we use this technology, but in a protocol is set up in such a way, the temperature is maintained in such a way that it has to be 22 degrees to 23 degrees only. Then the homology donor template has to be in large quantity. So you have to give it the concentration, you have to give like 3 times, 3:1. Like HDR should be 3 and 1 should be your gene which is being digested, where the gene that is being targeted, that is one, and but the HDR quantity has to be 2 or 3 times that quantity. So that all aspects you have to standardize. We have standardized it very well. So
okay,
we
okay,
get around
nice,
25% success. Every
okay,
100 plants, we get 25 plants are edited. So
mm,
this is that, yes.
okay. I asked just because in many papers I found that only 2 to 3% SDR absences are there, so that's why I asked this question. Okay, thank you for answering. Thank you.
Moreover, we use bialistic technology. So
Okay.
difference is that they may be using agrobacterium or some other.
Mhm. Okay. Thank you.
Thank you, Dr. Manish, and thank you all. Thank you, ma'am, for this beautiful lecture and quite good discussion. And we have another 2, 3 questions which, after being uploaded on YouTube, I will send it to you, Dr. Rajkumar, and one professor from University of Allahabad who is working in this field. He said that after watching your video, then he will send some questions and queries. I will share to you. Thank you all for joining this KSAS talk. And next month we will also come with new speaker and new idea to enhance the society. Thank you. Thank you very much.
Thank you so much, Alokji. Thank you,
Namaskar.
Balram.
Namaskar.
Thank you, Raj, Dr. Raj. Thank you, all of you. Thank you.